Spermatogenesis is the process of formation of haploid spermatozoa from diploid primary germ cells. So first of all, let us see that where the germ cells are located. So first of all, I am going to draw the structure of testes here so that we can understand easily that where these primary germ cells are located. So this is our testes, and we know that in one testis there are about 250 testicular lobules are present. Suppose these are our testicular lobules, and we know that in each testicular lobules there is about 2 to 3 seminiferous tubules are present. So suppose these are our seminiferous tubules, and in the inner lining of each seminiferous tubule there is about in the inner lining of each seminiferous tubule there is germinal epithelium is present. So suppose this is our germinal epithelium, and we know that germinal epithelium is a tissue, and we know that every tissue is formed of some cells. So this very germinal epithelium is formed of primary germ cells. So this is the place where primary germ cells are located. Now let's come on our main topic, that is formation of haploid spermatozoa from these primary germ cells. So I'm going to draw here one primary germ cell so that we can understand the process of spermatogenesis easily. Suppose this is our one primary germ cell, and it has its nucleus here, and it has its nucleus here. So we know that there are lots of primary germ cells like this, and it undergoes mitosis. So it undergoes mitosis to form two spermatogonia. I'm going to show you. So this is our spermatogonia. These are our two spermatogonia having their nucleus. And because there are lots of primary germ cells in the germinal epithelium, so these primary germ cells undergoes mitosis to form a lots of spermatogonia, and some of these spermatogonia act as stem cells. Stem cells are those cells which divide rapidly but do not undergoes growth phase. So because of this there is no menopause in the human males, and because of that sperms are produced in the male until the death of that very man. So this is our spermatogonia, and some of these spermatogonia act as other cells which undergoes growth phase. So now let us see that what happens when this very spermatogonia undergoes growth phase. Suppose this is our spermatogonia having nucleus. So when this spermatogonia undergoes growth phase, then it forms primary spermatocyte by a process of spermatocytogenesis, and in this process there is accumulation of nutrition in the cytoplasm, and the nutrients are absorbed from the other germ cells, and there is also replication of DNA. So when the replication of DNA is completed, then this very spermatogonia is said to be primary spermatocyte, and we know that when there is replication of DNA, then there will be 100% surety that this cell will undergo cell division, and here the cell division which take place is known as meiosis. So now let us see that how meiosis will take place in primary spermatocyte. So I am going to draw here the primary spermatocyte, and it has its nucleus here, and in the nucleus, because there has been a replication of DNA has taken place, so there is going to be the two chromatids. So these are the two chromatids, and the two chromatids are known as chromosomes together. So these are the two pairs of chromosomes. These are the two pairs of chromosome. Sorry, this is one pair of chromosome. This is one pair of chromosome, and as like it, there are 23 pairs of chromosomes in a human diploid cell. And so, this cell, this very primary spermatocyte, undergoes meiosis 1 to form two secondary spermatogonia. Sorry, spermatocyte. This very primary spermatocyte undergoes meiosis 1 to form two secondary spermatocyte. So now let us see that how meiosis 1 will take place. So in meiosis 1, this will give rise to two secondary spermatocyte having their nucleus, the nucleus is made with the same color. And so in meiosis 1, this very pair of chromosome is going to divide like this, and because of that, in this cell there is only 23 chromosomes, not the 23 pairs of chromosomes. There is only 23 chromosomes are present in this very secondary spermatocyte, and as like it, there is also only 23 chromosomes having the two chromatids. Now I am going to divide it again. So suppose this is our secondary spermatocyte and it has its nucleus here and it has the two chromatids here. So because this very cell, that is secondary spermatocyte, has only 23 chromosomes, not 23 pairs of chromosomes, there is only 23 chromosomes because of meiosis 1. So this very cell is undergoes meiosis second, and in meiosis second this will form two cells with 23 chromosomes, and it has its nucleus here and it has 23 chromosomes. So because this is only one, this cell is only one secondary spermatocyte, and we know that there are two secondary spermatocyte is formed from one primary spermatocyte. So again there is another division takes place in the other secondary spermatocyte, and because of this there are four haploid cells are going to produced from two secondary spermatocyte and from one primary spermatocyte, because one primary spermatocyte gives rise to two secondary spermatocyte because of meiosis 1, and these two secondary spermatocytes will give rise to four haploid spermatids because of meiosis 2. So these are the two spermatids, and as like it, there are also two spermatids are also formed. So these are the four spermatids are formed, and this process is known as spermatidogenesis, that is the formation of spermatids from the secondary spermatocytes is known as spermatidogenesis. Now we see that these four spermatids are circular and they are non-motile and non-functional. But we have already learned that sperm is a motile cell. So how these spermatids gain motility? We have to discuss about that. So the transformation of these four spermatids, there are not only four spermatids are formed, there are lots of spermatids are formed, but there is the formation of four spermatids from only one primary spermatocyte. So the transformation of spermatids into spermatozoa, that is the transformation of non-motile non-functional spermatids into functional motile spermatozoa is known as spermiogenesis. So now let us see that what happens in the process of spermiogenesis. So in the process of spermiogenesis, the circular spermatid is going to look like this. This is the plasma membrane of the spermatid, which transformed like this, and it looks like this. You could recall the structure of sperm. So the plasma membrane forms the structure like this, and the Golgi body of that very spermatid form the acrosome of this very spermatozoa, and the nucleus is here. Here is the nucleus, and in the nucleus there is a deep fold inside it. I'm going to show you. So here is the deep fold inside the nucleus. So here is the deep infold inside the nucleus. Sorry, I'm not so good in drawing, so I have such problem. So suppose there is the infold of this very nucleus, and one of the centriole of a pair of centriole is placed here. So one of the centriole is placed here, which is known as proximal centriole, and the other centriole is in the middle piece, which is known as distal centriole. So the proximal centriole forms the spindle during the zygotic division, and the other centriole, that is distal centriole, will form the axoneme. So the axoneme are formed from this very distal centriole. Axoneme is basically the axial filament, and the mitochondria of that very spermatid, we here discuss the transformation of that very spermatid in the spermatozoa. So the mitochondria of that very spermatid will form a spirally coiled sheath around the distal centriole and the proximal part of axoneme, and it forms a spirally coiled sheath like this, which is known as Nebenckern. So Nebenckern is basically the spirally coiled sheath of mitochondria of spermatid. And the cytoplasm of spermatid, 80% of the cytoplasm of spermatid get lost, and some of the cytoplasm, that is the remaining 20% of cytoplasm, will form the sheath of tail. So that will form the sheath of this very tail. I'm not going to show you the sheath of that very tail because sheath is in the tail here. So this is basically the process of spermiogenesis. So after spermiogenesis, this spermatozoa becomes functional and motile. And now we discuss all these processes taken place in the germinal epithelium of the seminiferous tubules of testicular lobules of testes. So when there is erection in the penis, this very spermatozoa is going to release from that very germinal epithelium into the lumen of seminiferous tubules, and before the release of this there is also maturation of sperm, and in maturation there is a accumulation of nutrition from the Sertoli cells of seminiferous tubules, and that process is known as spermiogenesis. gonia, spermiogonia. So this very process is the whole process of spermatogenesis. That's all. Thank you.